Mendel, born in 1822, is considered a father of genetics, whose experiments made our understanding of genetics today possible. He experimented with many plants to develop laws of inheritance. He settled on pea plants as his main experimental plant. He chose pea plants because they grow quickly and are easy to breed and have a variety of obvious traits. In 1865, he took his nearly 30,000 pea plants to display at the National History Society, and he demonstrated that traits are transmitted from parents to offspring independently of other traits and in dominant and recessive patterns. After this, he went on to publish a book, Experiments in Plant Hybridization, which went unnoticed by scientists at the time, thinking it was wrong. We know now that this is not the case.
Gregor Mendel's experiments, while foundational to modern genetics, are often surrounded by misconceptions. Contrary to popular belief, his work wasn't immediately recognized as groundbreaking, and he faced challenges replicating results with certain plants. Mendel didn't use the term gene. Instead, he referred to the genes as factors. The notion that his experiments were perfect has been debunked by statistical analysis. The order of his experiments, as presented in his 1866 paper, may not reflect that actual chronology, and his results were carefully selected examples rather than random data. Not all traits he studied segregated independently. As previously thought, it's also a misconception that Mendel fully grasped the broader implications of his work for heredity. His focus was primarily on hybridization. Lastly, the universal application of Mendel's laws to all organisms is an oversimplification, as demonstrated by his confusing results with hawkweed due to its unique reproductive process.
The first work of Mendel we will be discussing are the laws of probability.
Mendel's laws of probability talk about how while crossing F1 plants, the traits occur in a 3-to-1 ratio, where the dominant trait comes up 75% of the time, while the recessive trait only pops up 25% of the time. Here's an example of this using the Punnett square.
So this is the Punnett square. This is what Mendel used to check the probability. So the capital T's are the dominant traits. So those are the ones that you'll see whenever if these are the ones that you get, the capital T is going to be the outcome, like what you see. The lowercase t's are the least dominant, so you won't see those if they have a capital T next to them. But if they have 2 lowercase t's, then you'll get the dominant or the least dominant trait. And so like these are called phenotypes. Or all 3 of them, or 4 of them, are called phenotypes.
They have the same phenotype.
Huh?
They have the same phenotype.
Yeah, these 3 have the same phenotype. This is a difference. So it's a 3-to-1 ratio. And then these 2 are different, but these 2 are the same. So their genotype would be a 1-to-1 ratio. So you would have 1 chance to get both dominant, 2 chances to get dominant and least dominant, and then 1 more chance to get least dominant. So 25% for these 2 and 50 for these.
Mendel discovered this by breeding pea plants. In fact, he bred nearly 30,000 pea plants, as said in the introduction.
The forked line method is a genetic tool used to determine the phenotypes and genotypes of offspring when there are multiple crosses. It avoids the complexity of a giant Punnett square. When there are multiple crosses, a Punnett square can get huge. For example, it could have a grid containing 256 squares. By using the forked line method, it organizes probabilities using branch diagrams. This is best used for analyzing multiple traits. Here's an example of the forked line method. As you can see, when you're combining 4 genes, it can get very confusing.
X-linked recessive inheritance is a pattern of genetic information where traits or disorders are passed down through the X chromosomes. One notable example of an X-linked recessive trait is red-green color blindness. The condition affects the ability to distinguish between shades of red and green. Though visual acuity remains normal, red-green color blindness is more common in males than females due to an X-linked nature. Males only need one copy of the defective gene on their single X chromosome to express the trait, while females require 2 copies, one on each X chromosome, to be affected. The inheritance pattern of red-green color blindness follows a specific rule. Affected fathers cannot pass the trait to their sons, as sons inherit the Y chromosomes from their father. All daughters of affected fathers will be carriers of that trait. Sons of carrier mothers have a 50% chance of inheriting the condition. The X-linked recessive trait demonstrates the characteristics skipping generation patterns where an affected grandfather may have an unaffected grandson through his carrier daughters. If a plant has 2 dominant alleles for flower color, it will have violet flowers. If it only has 1 dominant allele and 1 recessive, it will show the more dominant allele. Only when the flower has 2 recessive alleles, it will show the recessive traits. We used the Punnett square, which would help show the probability of the traits, and it shows how the alleles will combine. We usually see a 3-to-1 ratio in a hybrid crossing, but sometimes we don't know whether the trait is homozygous or heterozygous. We can test to find out which one is which by crossing this organism with one that is homozygous or recessive. If any of the results show the recessive trait, then we know the original organism was heterozygous.
Mendel also used probability in his pea plant project. He used a table to find out the percentage chance of a trait being dominant or recessive. This can be shown by using a dice. Using a dice, you can say that numbers 1, right here, or 2 are the recessive traits, while all the other numbers, 3, 4, 5, and 6, are the dominant traits. We're going to roll the dice a couple of times to see the different chances of it landing on the other. As you can see there, that's a 1. So while there's a less chance of it being a dominant trait, it still is possible for it to be recessive. We'll roll it again. This time we land a 4. So this is a dominant trait right here. And we're gonna roll it a couple more times and see what the final result is. This time we got another 2. So this is another recessive trait, which is this is actually uncommon. Usually you're gonna land more dominant traits. So now it is 2 dominant to 2 recessive. So we're gonna roll it 3 more times and see what the numbers are after that. A 6. So that is another 1 for the dominant. So now it is 3 to 2 in the favor of the dominant. Now we got 2 more rolls to see what we can get after that. A 5. So as you can see, as we roll more, it gets closer to the probability. We just need more numbers to make it accurate. And as we keep on going, it's been really dominant. Our final roll now to see what we finish off with. So as you can see, the final results ended with 5 on the dominant side and only 2 on the recessive side. This is what proves that Mendel's theory about dominant being more dominant was correct if you have the numbers to use. And that's it for this segment. Peace.
Crossing over is the exchange of genetic material between non-sister chromatids of homologous chromosomes during the prophase 1 of meiosis. This process happens when chromosomes pair up and swap the DNA segments at a specific point called chiasma. The outcome is a recombinant chromosomes with new genetic combinations of the alleles. This enhances genetic variation, which would help the population adapt to changing environments and prevent harmful diseases and viruses get any worse. Usually this happens 2 to 4 times per chromosome arm. Incomplete dominance is a genetic information pattern. This occurs when neither alleles appear as dominant. For example, if a dominant red, capital R, and a white recessive, lowercase r and lowercase r, merge, and they create a pink, capital R, lowercase r. When these two genes merge, neither one comes out dominant. This creates incomplete dominance, and the genes merge. When two pinks merge, they create a phenotype. 1-2-1, red, pink, white. This expresses the Mendelian genotypic expectations.
Sometimes, depending on the gene, certain characteristics will be coupled and knocked out by the characteristics of other genes. It's called epistasis. Epistasis is a genetic phenomenon where the expression of one gene is influenced, modified, or matched by one or more other genes, known as a modifier or epistatic gene. The interaction impacts phenotypic outcomes, such as a person's hair color and albinism. Where certain genes override others to determine traits and diseases, as it highlights nonlinear actions between genes with genetic patterns. Look here. You have 2 people. One person has 2 brown hair traits, which gives them brown hair. But another person has a brown hair and a red hair trait. This gives them red hair. That's all.
Lethality in genetics. Some alleles, when developed, are deadly. They are mutations in essential genes. Recessive lethal alleles are lethal only in homozygous form. Dominant lethal alleles are alleles that are lethal even in heterozygous conditions. Alleles are synthetically lethal when mutations in 2 genes disrupt pathways. Mendelian inheritance patterns are altered by lethal alleles.